Viruses. Why do we want to know about viruses? Well, again, the more we know about the pathogens and how they attack us, if you will, the better we can do for our patients. So we need to learn more about the structure of viruses so that we can protect ourselves. And this is obviously apparent right now because we are dealing with the after effects of SARS-CoV-2, which is the virus that causes the disease COVID-19. So we're definitely interested. So just take a second and I want you to think about how many viruses that you know of that causes diseases or a disease that's caused by a virus. So just take a second and do it. So there's a lot of viruses out there that cause disease, a lot of case studies caused by viruses. So again, the more we know about them, the better we can do. So let's talk about what a virus is. Okay, before we do that, how do we know viruses exist? Well, we can see them. We have an electron microscope that we can actually see them, so we know they exist. The electron microscope wasn't actually invented until the 1930s. How do we know about them before then? If we go back into history, if you remember chapter 1, remember that they were all looking for, after Pasteur put forth the germ theory. If bacteria, these microorganisms that Leeuwenhoek saw, if these microorganisms can spoil wine, can they spoil people? So everybody started looking for these microorganisms and their connection to different diseases at the time period. And that led to the field of study of etiology, what causes these diseases. So if you remember right, it was Robert Koch was the first person to match up Bacillus anthracis, a microorganism, a bacteria, to the disease anthrax. So took a lot of good research and scientific method on his part. He put forth the Koch's postulates after that, but it also took some luck because if he had been studying influenza, do you think he would have been able to figure it out? No, influenza, the flu, is caused by a virus. And so he would not have been able to figure that out because he would not have been able to culture it outside. Looking at bacterial diseases, there was a lot of matching that was taking place, but viral diseases were a little bit tougher. So there was some investigators in 1892, and again, they were looking for the cause of a disease. Now, this guy, Ivanovsky, and both of these guys actually were botanists. They actually studied plants. They weren't physicians, they were botanists, and there was a crop, tobacco, which was a big cash crop, just like the wine industry. And there was a disease that infected tobacco plants. And it was called tobacco mosaic disease. And it caused the leaves of the tobacco plant to turn brown and fall off, and the plant would be sick and die. So obviously that affects profits. That's a big deal. Now they knew this disease was infectious because they could take an infected plant that had this disease and they could rub the leaves on a healthy plant, and then the healthy plant would get the disease. So they knew that it could be passed from plant to plant. It wasn't something in the DNA or anything else at that time. They knew it was infectious. It could be passed from plant to plant. So they were looking for the bacteria, the microorganism that caused it, because so many of these were being matched up at this time period. So Ivanovsky was working in his lab and he was looking using Koch's postulates and he was trying and he could not find a bacteria that caused this disease. And finally, he took through, because he knew he could take the leaves of the plant and he could grind them up into a fluid, into a liquid, and that liquid was still infectious. And so he started passing it through filters and taking things out of the liquid, trying to find where that microorganism was that was infecting it. So he passed it through these filters and it remained infectious and it remained infectious. So finally he passed this fluid that was made from crushing up these sick tobacco plants. He passed this fluid through a porcelain filter. Now, porcelain was a porous material, but the pores in the porcelain were so small that bacteria could not pass through it. You could still get liquid to pass through porcelain, but not a cell, so not any bacteria. So he finally passed it through something that wouldn't let anything through except just a little bit of liquid, and that filtrate, that liquid, was still infectious. So he came to the conclusion there must be something smaller than bacteria that was causing this tobacco mosaic disease. And in fact, he found that there was something very small causing this. His conclusion, Ivanovsky's conclusion, was that this must be a really small bacteria that they could not see under the microscope. So that was his conclusion. Now, there was another scientist in another country working on the same project, and he heard of Ivanovsky's experiments, and he decided to repeat them in his lab. So again, two separate places, two different investigators. He wanted to repeat this experiment, which is a good thing. Anytime you can repeat something and get the same results, that leads validity to those conclusions. So Bergenic was decided to repeat that experiment. So he passed it through a porcelain filter. He got the same results. The fluid was still contagious, even though none of the cells made it through. Now, he was a little bit more forward thinking, and he said, I don't think this is a bacteria. I don't think this is a cell. I think there's something smaller than a cell that we have not seen before that's causing this tobacco mosaic disease. So because he felt it was something new, you get to name it. If you find something new, you get to name it. And so he named it Contagium vividium fluidium, after the contagious fluids, and vividium means poison from that. It got shortened because that was a mouthful to a filterable virus. And then eventually we just called it a virus. So he was the first person to realize that he was dealing with something new and give it a name. So before they could see a virus, they knew they were there based on its actions and what it could do. All right, so again, more work was going into it. In the 1930s, there was a biochemist that ended up purifying this virus and isolating it. And what was interesting about this is that when he isolated it, it crystallized. And cells, if you purify the cells, take out all the water, then the cells just die, right? You don't get crystals formed. Things that form crystals are your minerals, salts, minerals, geodes, those types of things. Inorganic material crystallizes. So this was kind of interesting, and they're like, again, I don't think this is a cell. I think this is something new. The electron microscope was invented, I said the 30s, it was invented in the 1940s, and allowed us to, instead of passing light and magnifying with light, we were able to pass electrons through an organism and then magnify it and see that electron pattern. This is how an electron microscope works. It means that we can magnify things much smaller than ever before. And so we got our first pictures of a virus. So this is a couple of different types of electron microscopes. This is a 3D image and some different viruses. This is both, I think it's smallpox. This one is Ebola. Has a different type of shape to it. All right.